Thank you very much, Matt, for agreeing to give a Build a Cell seminar, and you can get started.
Yeah, sure. Thanks, Kate, for the invitation. It's good to be meeting with you all via Zoom. So by way of introduction, I'm currently a professor of chemistry at Virginia Commonwealth University in Richmond, Virginia. I did my PhD work at the University of Michigan, working with a medicinal chemist enzymologist, Jim Coward, doing a lot of organic chemistry. And then I moved to the Shostak lab, then at Harvard for my postdoc from 2002 to 2006. And I learned a lot of molecular biology, and some of the work I did there forms the basis for what I'm still doing in my lab right now, which I'll tell you about today. So the thing I'm going to talk in particular about is extensive breaking of sense codon degeneracy in vitro. And so I'm used to giving talks like this in front of people who are all chemical biologists. I know this is a diverse audience, so I'm going to start a little slow and give some bigger background than I normally do. But if it's all familiar to you, or if you know it like the back of your hand, please bear with me. We'll get to the exciting science in a little bit. Okay. All right. So, the genetic code is shared by most all organisms, although there's some tweaks here and there in different archaeobacteria and other places. But, we have 64 codons mediated by the, let me turn my laser pointer here. Okay, 64 codons mediated by 3 positions with 4 nucleotides each. And they encode for 20 amino acids, start codons, stop codons. Just the first 2 positions are shown in this kind of nifty flower-like genetic code diagram. So what I'm going to be talking today is how can we work on breaking up this degeneracy? And so the genetic code is involved in the process of translation. And during translation, we take a messenger RNA message and turn it into a protein. And I'm going to give you a very simplified version of how that works in E. coli. So we start with a pool of amino acids, 20 of them, and a pool of tRNAs. In E. coli, there are 46 different tRNAs. These amino acids are charged onto the tRNAs by a class of enzymes called amino-acyl-tRNA synthetases. There are 20 of these, 1 for each amino acid, and they attach the correct amino acid onto the correct, or the cognate amino acid onto the cognate tRNA, giving you these amino-acyl-tRNAs. The attachment happens via an ester linkage, which can then be reactive. These amino-acyl-tRNAs then bind to a protein in E. coli, an elongation factor called EFTU. EFTU takes these proteins and takes these amino-acyl-tRNAs and delivers them to the ribosome, which is shown here in this teal structure. So in yellow is the mRNA, these quickly lines of the tRNA bearing amino acids, and the ribosome will read down the messenger RNA codon by codon by codon, each time bringing in an appropriate new amino-acyl-tRNA. And what the ribosome does is it catalyzes an amide bond formation between the nascent growing peptide and the new amino acid coming in. So an amide bond is formed, the tRNA switches positions. This is the A site, this is the P site. From the A site to the P site, a new amino-acyl-tRNA comes in, corresponding to the next codon, and the process is continued. And then eventually, at the end of the day, we make peptides or proteins as a result. Okay. So what enforces the fidelity here? There are really 2 places that enforce the fidelity of the genetic code. One is the amino-acyl-tRNA synthetases, and they enforce the fidelity of the genetic code by attaching the correct amino acid onto the correct tRNA. So for example, the synthetase that charges leucine will recognize both the leucine amino acid and all of the 5 in E. coli leucine tRNAs and attach the amino acid onto the tRNA, serving as a recognition element. There are some selectivity in the EFTU step, but not so much. So these leucine tRNA leucines are then ready to go in translation. And then what enforces the genetic code on the other end is the pairing of the messenger RNA and the tRNA. So here I've zoomed in and just shown you a particular leucine codon, CAG, and the tRNA that has an anticodon that pairs exactly with this in Watson-Crick fashion. So the ribosome can recognize it and bring this amino-acyl-tRNA in. Okay, so if we want to try to break the degeneracy of the genetic code, we're going to, and break the degeneracy of sense codons, we're going to have to manipulate this step putting amino acids on, and perhaps also what the ribosome can recognize. And the reason why we have to manipulate with the ribosome recognition step is illustrated by this somewhat complicated slide, but this is a kind of a tRNA-centric view of the genetic code. So this is a really nice paper by Alexandrov. We look here at a particular amino acid, serine, and these are for example, the 4 serine tRNAs in E. coli. And these arrows represent which codons each of these tRNAs read. And you can see right away that it's not one tRNA to one codon. That's why there are only 46 tRNAs in E. coli, but 64 codons, 61 sense codons, but 64 total codons. And so this tRNA, for example, reads the UCU codon and the UCC codon. This tRNA, which has a modified nucleotide here represented by a V, actually can recognize 3 different codons. And so if you want to try to break the degeneracy of the genetic code, you can quickly see we have some issues. We can't just take an individual tRNA and using some method attach a novel amino acid to it, the next tRNA a novel amino acid to it, and the next one a novel amino acid to it, because the tRNAs have these overlapping reading specificities. This, of course, is really important for life because it allows for redundancy. It allows for resilience against low concentrations of individual tRNAs because they have backups. And you can see as you look throughout the code, the serine codon is not representative. The leucine codon box is the one I'm going to focus most on in my talk today, and it has 5 different tRNAs reading these 6 codons with lots of overlapping specificity issues. So how do we get novel amino acids, additional amino acids into the code? Well, one way is to use sense codon reprogramming. And this actually doesn't expand the code at all, but it replaces an amino acid for another. For example, you can potentially replace the valine amino acid with something that's a valine analog. This work like this was originally pioneered by David Terrell and others, even in the 90s, where they would have an oxytrophic bacteria that couldn't make a certain amino acid. They would feed another amino acid in, a surrogate, and then they would look to see if that second amino acid was incorporated into proteins and place the non-canonical in place of the natural amino acid. But you're not expanding the code with this method. And the second method, which has been developed really extensively, originated with Pete Schultz, Sid Hecht, and some others, but really kind of brought to practice in particular by Pete Schultz and subsequent people who worked in his lab, are to use the fact that we have 3 codons that don't encode for any amino acids at all, the 3 stop codons. And if you can create a aminoacyl tRNA in an organism and add it to the organism, then you could perhaps re-encode one of the stop codons with a non-canonical amino acid. And in fact, this allows you to get up to 21 amino acids. And in fact, there's a paper now where people have re-encoded all 3 stop codons with non-canonical amino acids. So in principle, that can get you up to 23. But the real money, I would say, is if you want to introduce interesting novel amino acids into the genetic code, you really need to break up the codon degeneracy. There are 61 sense codons, but they only encode for 20 amino acids. And so if you could take, say, the leucine codon box, which normally has 6 codons here that encode just for a single amino acid, and convert it to something that, say, encodes 3 amino acids, now you've achieved a significant amount of expansion on a very narrow piece of the code. And if you can start to do that with all of these other coding boxes, you can start to find things that are pretty interesting. And maybe you could get up to around 30 amino acids. Why do we want to do this? Well, this is why my lab wants to do this. We actually harness the genetic code system to make diverse peptide libraries for drug discovery. And the diversity is dependent upon how many amino acids we can put at each position. So if we had a library that has 6 random codons in it, random amino acids, that could allow us about 64 million different peptides. If we could turn this into 40, that could allow us to get to billions. The technology that my lab uses that I'm not going to talk about today is called mRNA display, and we can make even tens of trillions of unique variants of peptides, macrocyclic peptides, from which we can then find ligands to interesting and novel proteins. So we are interested in, for this reason, But there's also a lot of people who are interested in this, but through creating novel expanded codes. Some of my favorite work in this area has been done by Chin. And what they have done is they've taken the standard E. coli, they've completely re-engineered the genetic code synthetically and deleted certain codons. So here's the 6 serine codons here, and they've actually re-encoded E. coli, deleting 2 of those codons altogether. So they're not present anymore in the genome. That then frees you up to start to put novel things in. Of course, it doesn't completely free you up because the tRNAs are still there. So if you delete the codons, but still have the tRNAs, if you try to put something else in, these tRNAs are going to go back and read these 2 codons, and you'll be where you are. So what they've done subsequently and accomplished last year in science was they deleted the tRNA genes. So the organism doesn't actually need the CRU and CRT tRNAs anymore if it doesn't have the codons that can be read by those tRNAs. And so now they're deleted. And so now they have an organism with only 4 serine codons, and that leaves 2 that are empty. And they did something similar with the stop codons. And then they introduced novel amino acyl tRNA synthetases and novel tRNAs, and they could re-encode these with some novel non-canonical amino acids. Very interesting paper in Science. So at the very beginning, the fundamental aspect of this, one thing that is particularly interesting is how do we get these non-canonical amino acids onto the tRNAs? Of course, you can't feed any non-canonical amino acids into translation apart from a tRNA. And so there are 4 methods. In one method, you can just use the amino acyl tRNA synthetases. Some work I did as a postdoc showed that the wild type synthetases are fairly promiscuous towards non-canonical amino acids. In addition, engineered synthetases can be used for producing and introducing novel amino acids. Similarly, you can use an RNA catalyst called a flexozyme. This only works in vitro. Hirosuga developed this flexozyme catalyst. It allows you to take an activated amino acid and using an RNA catalyst, attach it onto any tRNA. Very generally, hundreds of different non-canonical amino acids have been used. It's a very powerful technology. The old school method is to create a tRNA that's missing a couple of nucleotides at the end, and then you synthesize an amino acid bound to those last 2 nucleotides, and then using RNA ligase ligate them together to make your synthetic tRNA. And finally, you could potentially take a natural amino acid, charge it onto the tRNA, and then chemically derivatize it using some sort of novel chemistry to get it here. All these methods have been used to create amino acyl tRNAs with non-canonical amino acids. I'm just going to focus my talk only on this left-hand side here, and particularly looking just at standard amino acyl tRNA synthetases. So we could do all this and do all this work in vivo, but that becomes complicated because keeping organisms alive when you manipulate and mess around with their genetic code is pretty tough. And so we actually, and for the rest of this talk, all of the work that I've been doing has been using a reconstituted translation system called the Pure system. It's commercially available, but the commercial system doesn't allow you to really control things very well. So we have our own version of this in my lab. It's a lot of work to prepare and get this together. So it requires you to make 30 different proteins, all 20 amino acyl tRNA synthetases, all of the initiation, elongation, and release factors, ribosomes, energy cycling system, mRNA. So the stuff in the box we make, as well as the ribosomes, we don't make total tRNA, we purchase that. And the power of this system is that we don't have to keep an organism alive to manipulate with the genetic code, and we can easily leave out things and reintroduce things in their place. So other people have been using this pure translation system. This is a nice paper from last year from Bristol-Myers Squibb, group at Bristol-Myers Squibb, where they actually have taken this pure translation system. Here's the standard genetic code. And by isolating individual tRNAs and introducing them, they've been able to break apart the genetic code to encode here for add 6 non-canonical amino acids to the code. One of these was introduced only at the initiation codon, but the other 5 were introduced throughout their code. And so the way they did this is they looked at individual codon boxes. So for example, these two codons right here, or these 4 codons right here, encode for serine, and using some careful planning and looking at individual tRNAs, they were able to split this serine codon box to encode for 2 different amino acids. Similarly, the 3 arginine codons here, and here, the 6 arginine codons, they were able to split into 3 amino acids. And this is pretty nice and impressive, but these codons actually have been carefully picked based on the known tRNA recognition overlap rules. And so to get beyond that is kind of challenging to think about how to do. Still, it's very interesting and very nice work. And these are mass specs using individual mRNAs that encode for some of these amino acids. And you can see they get pretty clean spectrum. There might be some orthogonality issues here with little blips. And the problem is really illustrated like this. We would love to be able to take this leucine codon box, which has 6 different codons in it, read by 5 different tRNAs, and we'd love to eliminate all these overlaps so that we could actually encode maybe 5 different amino acids with these 5 leucine tRNAs. So we went ahead and started trying to study this. And one of the things we thought about right away is we really want to verify if these codon reading rules are right in vitro using our translation system. And to do that, we needed a system to be able to easily and quickly isolate individual tRNAs from the total tRNA mixture. And so my student Cal McFeely developed a novel fluoresce capture strategy for tRNAs, and this really would work for any RNAs. It's not selective to tRNAs at all. And the concept here is that fluoresce affinity. So if you take a fluorocarbon tail and append it to a molecule, it will have high affinity for a fluoresce resin. So what my student Cal did is he made a few different versions of oligonucleotides complementary to a given tRNA, shown here in these green dots are the sequences he tried. He tried things that span the anticodon, this variable loop, things that span the D loop. But each of these oligonucleotides have fluoresce ponytail. And so the concept was you throw this oligo in with the tRNA mixture, you denature it, you cool it back down, and because that oligonucleotide is complementary to that given tRNA, it should anneal. Then you can pass this through a fluoresce affinity column. So you can wash this extensively, and then you can elute using a strong organic solvent, break the fluoresce interaction. Actually, break the tRNA. Sorry, I misspoke. You elute actually by denaturing this by heating it and using aqueous conditions, low salt, to pull the tRNA off. You then actually can regenerate the column and pull the fluoresce oligo off in a separate step. So the beauty of this system compared to biotin and other options here is that the hybridization happens in the liquid phase. So this fluoresce oligo binds to the tRNA, so you don't have to worry about columns yet or beads yet. And then you pass it through a column. These fluoresce columns can be reused dozens of times. So unlike a biotin-related oligo, where it's stuck on those beads, and those beads have to be used for that purpose from then on because it's hard to break that interaction. In this case, we can reuse these columns with different fluoresce oligos every time. So Cal was working on developing this technique, and he created oligos for a number of different tRNAs. And this is what commercial total tRNA looks like. And this is what our oligos look like after capture. You can see here that we typically have one major band. Interestingly, in a lot of cases, particularly these leucine tRNAs, we get a second band. Those second bands turn out to be tRNA fragments. We showed that by moving the position of the oligos, and we showed they weren't captured anymore. So we were able to optimize this. I'm going to focus here on the leucine tRNAs. And so these are the wild type leucine tRNAs. You can see here they are one band. We also made the in vitro transcribed versions of these using in vitro transcription. These lack all the post-transcriptional modifications. Just to check that they were pure, we did northern blotting. So we took our individual leucine tRNAs and we looked to see if any other tRNAs were present in the mixture, and we can see really get nice clean northern blotting. We also took these amino acyl tRNAs. We put in all 20 amino acyl tRNA synthetases and all 20 amino acids, and we have a mass spec assay that clips off the end amino acyl amino acyl AMP and gets a mask. And you can, these masses, the peaks here all correspond to leucine. So we're not seeing issues with other tRNAs being present. Some little blips here and there, but these are predominantly pure species. So using this technology, we can now take and capture individual wild type fully modified tRNAs from E. coli total tRNA and compare them to their in vitro transcribed counterparts. So here's what we're doing. So we take these wild type tRNAs and we decided that we wanted to compete them against the T7 tRNAs. So we created these competition assays just to see which was better and more efficient at reading a given codon. So we'd pick a given codon, we make an mRNA, and we take leucine and charge it onto a given, say, a T7 tRNA. And then we'd actually charge a deuterated version of leucine onto the wild type tRNA. We put these two together in equal concentrations in a translation reaction. We let the translation go. We would capture the resulting peptide via affinity chromatography, and we would measure the intensity of those two peaks. And perhaps not surprisingly, we see that the wild type tRNAs win in every case over the in vitro transcribed tRNAs. So these modifications are actually important in terms of translational efficiency. We then took this a step further, and we looked at putting non-canonical amino acids onto these individual tRNAs. So we looked at a couple of different re-encoding schemes. This is a pretty busy slide, but it's getting into the nitty-gritty details is not so essential here for this talk. But we cherry-picked our tRNAs based on their codon reading preferences. We charged them either with all leucine, all right, and so that gives us a single peak here, single peak in both of these mRNAs. Nice single mass spec. We got 3 leucines. We then put non-canonical amino acids in there. So we took leucine. One of these is a T-butyl, one is a fluorinated version of leucine. You can see the structures of those two here. And we do a translation, and using the wild type fully modified tRNAs, we can actually see we get one predominant peak. This is the diamond here is the correct peak, but we do see some little blips. If we look at these particular tRNA, we get something that's a little cleaner. Interestingly, if we charge the T7 tRNAs here, we start to see orthogonality breakdowns. So these tRNAs that have the modifications are actually better at reading their codons than the T7 modified ones. Interestingly, in this case, this particular tRNA, T7 tRNA, doesn't read its codon at all, or only a little bit, and we actually get a methionine misincorporation there at the UUG codon. So we've shown here that we can isolate these tRNAs. We can start to use them to break apart the genetic code. But really, there are some problems here. And that is, these tRNAs were cherry-picked because they should be the only tRNAs that read these codons. So based on the literature, this tRNA LU2 should be the only thing that reads the CUC codon. This LU3 should be the only thing that reads the CUA, and this LU5 should be the only thing that reads the UUA. But when we do the translation experiment, we get a major peak, but we get lots of other little peaks here, showing that even in the situation where we think it should be the very best orthogonality cherry-picked, we're getting overlapping reading. And that is even harder when we start to put non-canonical amino acids on these tRNAs, which may have slightly different efficiencies. Let me take a drink for a second. All right. So we went about trying to solve this problem. And what we realized is that we didn't just need this tRNA reads this codon, this tRNA reads this codon. We needed to be able to rank the efficiency of a given tRNA for reading a given codon. And so we created a similar competition assay that you saw before, except in this case, we are now putting all 5 tRNA leucines here together in one pot, but we've charged them with leucine analogs with different mass. And so we now can actually compete all 5 of them at once for a given codon, and we read out the mass spec, and we look at the heights of the peaks, and that tells us what the ratios are. In other words, which tRNA won that codon, and how well did it win that codon. And before we did that, we wanted to really make sure that we were able to create balanced amino acyl tRNA mixtures, so they really were 1 to 1 to 1 to 1 to 1 when they were competing for the codon. So we used our mass spec assay, and our tRNA mixtures. So this is what the mass spec assay looks like. We get these really nice tRNA mixtures. And they should be 20% or all around, but they're pretty close. We also used in vitro transcribed tRNAs, and we see the same. We see the same thing. So this verifies that we're putting the amino acyl tRNAs in the translation at the same concentration. And now we're off to the races in testing these codons to see which tRNAs win. And the situation was actually worse than we thought when we first started this project, because we looked at our fully modified tRNAs, and what we see is that the black boxes are the things that have been reported in literature to read. These tRNAs should be able to read those codons. And we see, troubling trends if we're trying to break the sense of codon degeneracy. So the CUG codon, for example, is read 53% by this tRNA, 24% by this tRNA, and 15% by Leu4, which isn't even supposed to be able to read that codon. The CUA codon was the most surprising to us. It's supposed to only be read by Leu3, but actually it's not only read by Leu3, it's also read by Leu2 and Leu1, almost in equal proportions. So this was a fairly discouraging result if we're trying to think about how to break apart the degeneracy of the genetic code, because it tells us that there are a lot more lines on those overlapping codon reading charts than have been really reported in the past that we've now uncovered with our competition assay. We looked at the individual in vitro transcribed tRNAs, and the situation is a little better. So it turns out that some of these modifications actually allow the tRNA to have a broader codon reading specificity. And here, you're looking at this heat map, look at the CUG codon. Now we've improved from 53 to 66%, a little better. The CUC codon is looking pretty solid, 93% read by Leu2. CUU is also looking pretty solid. CUA codon is still a problem. These UUG and UUA codons are looking a lot better with in vitro transcribed tRNA. So even though in vitro transcribed tRNA failed in the previous approach, this shows that by choosing the right codon and by not competing wild type and in vitro transcribed together, we can really improve orthogonality. But still, this is not great. If we really want to create a nice, crisp orthogonality between codons and tRNAs, we're not going to be able to get away with 48, 19, 28, or 66%. So at this point, my lab was actually working on, in a completely different project, was working on developing hyperaccurate ribosomes. These are ribosomes that have been isolated in response to streptomycin resistance. Decades ago, they were isolated. And it turns out that the way these hyperaccurate ribosomes achieve their hyperaccuracy is that in this accommodation step, occasionally the ribosome will make a mistake and it will bring in a near cognate amino acyl tRNA. And if it brings in a near cognate amino acyl tRNA, sometimes it gets rejected. Usually it does, but occasionally it will actually get accommodated and brought in incorrectly. And you can measure this via a variety of different strategies. So these hyperaccurate ribosomes actually, the equilibria here, this accommodation and rejection step are altered. And so it makes it harder for a near cognate tRNA, amino acyl tRNA, to find that codon and get in and move into translation. So we were testing these at the same time as we were doing this project in our in vitro translation system. This is just one of the figures from our paper where we tested them, and just showing you the power of these ribosomes. So here's a full length, here's an mRNA that we designed. We put a his tag at the N terminus and at the 5' end, the N terminus of the peptide, and a flag tag at the 3' end of mRNA, C terminus. And so we just did a bunch of translation reactions with wild type versus these mutant S12. That's what I'm going to call them, because that's the protein that's mutated ribosomes that are known to be hyperaccurate. Interestingly, we also decided to challenge the ribosome by putting either, in one case, the least challenging case, only the amino acids and synthetases required to create this template. And then in another case, only we put in all 20 amino acyl tRNAs. And what you can see, this is just measuring yield of, so we took the translation, we split it in half, we captured with his or flag. So any his captured yield just means that it's only made it through this far in the translation correctly. And the flag capture means it's made it all the way through. So you can see, if even just with wild type ribosomes using the minimum synthetase, we see a drop off in yield between His and flag. And that means that the wild type ribosomes are not always making it to the end. Interestingly, if we look at the mutant S12 ribosomes, we don't see a significant difference between the His and the flag, which tells you they are processing all the way to the end. Another interpretation here is that these ribosomes could be making some errors, which might reduce affinity, and that's why you see the difference. Then when we start to put in all 20 amino-based tRNA synthetases, we can see here that if we look flag to cis for His to flag ratio here for the wild type ribosomes goes down even further. Even though the yield has dropped for the mutant S12 ribosomes, we can see here that they still don't care. Even with all these unused amino-acyl tRNAs around, that could be potentially challenging the ribosome as neurocognates. If we look at the mass specs of these reactions, we can see here that in the case where we just add the minimum synthetases, we see nice correct peaks for both His and flag capture. But when we put the wild type ribosomes in there with all 20 enzymes, we start to see erosion in yield, but the mutant S12 ribosomes are looking just fine. This is something we've noticed for a while. People don't typically report it when they're doing translations in these particular systems, but if you look at a lot of papers, typically only the required amino acids and tRNAs are added. So this got us thinking, well, if these ribosomes are able to discriminate against near-cognate amino-acyl tRNAs, maybe they actually can discriminate with tRNA isoacceptors within a given codon box. And so we started to work on this, right? So this is the plot I showed you before with wild type ribosomes. You can look at the numbers here. And when we switched from wild type to hyperaccurate ribosomes using our same competition assay, we actually see some improvements. Most notably here, the CUA codon goes from 36% up to 66%. So that means that these hyperaccurate ribosomes are actually discriminating better between Leu2 and Leu3. Remember, these are cognate tRNAs for a given codon. Okay. And we see some improvements all across. So this was looking promising, and we were kind of excited about what this might hold when we looked at the T7 case. So these are in vitro transcribed tRNAs that look like this before, and now this is the pattern that we see with in vitro transcribed tRNAs, where we can really see individual, highly pure, not complete orthogonality, but very high orthogonality across all of these leucine codons. We'd love to see these over 90%, but still, this is quite solid. And so now we wanted to see, all right, can we really, since we have these new rules showing that we have a predominant tRNA for each of these 5 leucine codons, what can we do with them? Well, we created a single template now that has all 5, has 5 of the 6 leucine codons. We use a single template. We do a translation reaction. And the question is, do we get lots of peaks corresponding to these deuterated leucines, or do we get a single peak? And this is what our translations look like. Really, we did this in triplicate. We saw one predominant major peak. I don't have the blowups of these, but if you look at the isotope reading patterns, they match what you would expect for the ability to break apart the 6-fold degenerate leucine codon box into 5 different amino acids. Now, of course, this is the simplest and easiest case because these are all leucines. And so once you start to introduce non-canonicals in there, you expect the situation to get a little bit more difficult. So we put non-canonicals on to these individual leucine tRNAs. We made this particular peptide using some different enzymes to charge these onto the tRNAs. And after a little bit of optimization, in particular, this benzylhistidine tRNA needed to be elevated in translation from our standard 15 micromolar up to 30. But after a little bit of optimization, we really get clean peaks. So now we are introducing 5 different amino acids, leucine plus 4 novel non-canonical amino acids into these, into the leucine codon box. And the peptide that we made is shown here. So the next question was, well, can these hyperaccurate ribosomes start to break apart the degeneracy of other codon boxes? And so we looked down the table a little bit at Valine. So E. coli has 2 different Valine tRNAs that read these 4 codons, and this is the overlapping codon reading specificity. Val1 is particularly a problem because it's been reported by Alexander to be able to read all 4 of these codons. So we charged each of these 2 tRNAs with different valine isotopes, just like we did before. Here's the mRNA. This is the ratio of the amino acyl tRNAs that we added to translation, basically 1 to 1, a little bit off from that. And this is our codon read pattern. So we can see for the GUG codon, we have almost all completely read by Val1. The GUC codon is almost completely read by Val2. The GUU codon was not clean. We do have, 75-25 mixture or so here. And the GUA codon is nicely read by Val1. So these are some of the aspects that show pretty solid translation. There are some other small peaks here that show that their functionality isn't perfect. But still, other than this GUU codon, we've now investigated 10 different codons, and we've shown that there's a predominant tRNA for all 10 of those, for 9 of those 10. We then put non-canonical amino acids onto these different tRNAs based on that codon reading pattern. So either valine or this TB lysine analog, and to show that we can get clean translations. Some little blips showing not perfect orthogonality. We then wanted to assemble all this together. And so we now created 3 different mRNAs. Each of them had 3 codons in it, 1 from the valine codon box, 2 from the leucine codon box. So these are the 9 of the 10 codons represented. So we've now taken just this left-hand side of the codon table and put in these 7 different amino acids. And so what we showed is, if we looked at the yields of these translations, this is the yield of the translations where we had the canonical amino acids, and the relative yield dropped in all 3 cases for the non-canonicals. But in 2 of the 3 mRNAs, we got really clean translations. So here's mRNA1, gives us the expected mass. mRNA2 had a little bit of orthogonality. We see some issues here with the leucine codons. And mRNA3 also looked really clean. So although these results aren't perfect, they really show that by combining hyperaccurate ribosomes and these in vitro transcribed tRNAs, we are now able to start to break apart the genetic code and the degeneracy of the sense codons in ways that we really thought would be impossible before, based on this type of pattern. So with the leucine codon box, at least, we've shown that using hyperaccurate ribosomes and in vitro transcribed tRNAs, we're able to take this overlapping codon reading pattern and turn it into this. And of course, now we're working on investigating many other codons to see how well these hyperaccurate ribosomes combined with T7 tRNAs work, with the goal of maybe splitting the genetic code up, not just into encoding 20 amino acids, but perhaps maybe up to 30 or even 40 different amino acids by combining these 2 technologies. And I think there's a lot of interesting implications also in vivo if you start to think about how these hyperaccurate ribosomes could start to re-encode things and change tRNA reading patterns. All right, so this is my acknowledgement slide. So all the work that I talked about was done by 3 really talented graduate students. Cal McFeely did the majority of the work that I just talked about. He's now at Merck. Bipashna Shakya, she developed the hyperaccurate ribosomes and has a paper on that. And Chelsea is kind of followed along behind Cal and Bipa doing a lot of these tests. And maybe you will be the next to join this project. I've got a postdoc position available. Feel free to send me an email with your CV. I'm happy to investigate that and have a look. This work was funded by the Massey Cancer Center, which I'm a part of here at VCU, and also the NCI and NIGMS at the NIH. I'd like to thank you all for your attention. I am happy to answer any questions.
Thank you so much. That was fascinating. Honestly, it's kind of reassuring to me to see that you're seeing some of the problems that we've been seeing ourselves. It's makes me feel less alone. There is a lot of questions in chat. Can you see chat?
Yeah, let's see if I can see chat. Let me turn off my pointer here. Let's see chat.
If you could read a question out loud before answering it, that's going to be great, because people that watch the recording won't see the questions. So
yeah, okay, cool. All right, I stopped sharing because now I can more easily see the questions. All right.
Yep.
Okay. Okay. First question is by Tanner Mueller. When you do sense codon reprogramming, is it possible to pair any natural amino acid with any random tRNA, or are there any rules? Are some tRNAs working better with some amino acids than the other? That's a great question. So I completely ignored EFTU. I mean, I mentioned it at the beginning, but EFTU is an important determinant here. So the ability of EFTU to capture tRNA and deliver it to the ribosome is an important feature. Suga and others have shown that kind of uniform tuning of the EFTU amino acyl tRNA interaction improves this. So there are, I would say, to answer that question, I think you can probably put almost any amino acid almost onto any tRNA with a lot of optimization, get it to work. But by thinking about EFTU affinity, you can maybe streamline that process a bit. We've also shown that by elevating the amino acyl tRNA concentrations, we're able to get better translation yields. Okay, second question. Is it possible to engineer a hybrid tRNA with an anti-codon loop for one amino acid, but the rest of the tRNA body for another tRNA? Yes, yeah, definitely it is. Especially if you use in vitro transcribed tRNAs, you can put any sequence in there that you want. And in fact, Suga has done all of his codon engineering stuff based on a tRNA asparagine body. It's a mutated asparagine body, and he just changes the anti-codon to read different codons. So that can for sure be done. He charges all of his with Flexazon. So if you do that, obviously, and you're using enzymatic charging, you got to think about, does the synthetase recognize the anti-codon? What happens, if you change the anti-codon, will the synthetase not recognize it anymore? But if you're using Flexazon, you can get around that, and that's not a problem. All right, interesting talk. What is your thought regarding our 20 amino acid world, neither 19 nor 21? Evolution had the chance to diversify further beyond 20 or 21, including syncing. Oh boy, that's a great question. It's not something I've thought about. I don't tend to read those evolution papers, surprisingly, considering the postdoc lab I came out of. But I bet some other people in this group would probably have some better thoughts about that than I do. I think I can pontificate here. I think nature probably minimizes complexity. And if you look at the types of amino acids that are in the code, there is some redundancy, but a lot of the novel amino acids have unique functional groups that are really important in protein. But I guess maybe you could think about some of the hydrophobic ones being pretty similar, and hard to argue that. But it's not something I've thought a lot about. Interesting question. All right. Do you think some of the problems with orthogonality and recoding experiments could be because when you purify ribosomes, even impure, there are some endogenous amino acylated tRNA species coming through on the ribosome, and that contaminates your mixture? Great question. We have done translations where we've left out tRNA and they don't work. We get no yield. So I don't think, there may be some amino acyl tRNA around, but I don't think that's enough to really push translation to completion. All right. Do you know if the ribosomal fall off non-completion rate is similar in vivo? I should know the answer to that. I've looked at some of those papers. I suspect it's worse in vitro for sure, because we don't have all of the proteins that are really necessary that the E. coli has for translation in our system. For example, we don't have EFP in there. That's been shown to improve things with polyproline codons. So it's probably worse. People have looked. I think the problem is that most people don't look for it because they're making full-length proteins, right? And if it's not full-length, it's not active, and who cares, right? Even a lot of the stop codon suppression experiments, they're just looking for, can you read through a stop codon and make the entire protein? I'm trying to think of it in vivo experiment that would really, you'd have to do some sort of pull down, look at what all the products are, and that's kind of laborious. But maybe someone else has some ideas on that. Okay, next question. Do you think that there is a context dependence regarding codon tRNA recognition specificity? We're trying to figure that out. I think there probably is. There probably is. And that just is another layer of complication, but yes, probably. All right, well, let's see, last question. When you use non-canonical amino acids, are there any problems with protein folding, especially co-translational folding? Does the ribosome have any proofreading capabilities to detect wrong unnatural amino acids? Okay, we are just making peptides, so we don't expect to see them fold. We don't look at folding. In fact, they shouldn't fold. Although we do have some parts of my lab are working on trying to lock things into shapes with cyclization. So I don't have any comment on co-translational folding. I know that some people, when they make proteins in vitro, put in chaperone proteins to help fold things. So I think that can be an issue, but that's not something I've looked at with non-canonicals. It'd be an interesting thing to investigate. Let's see, does the ribosome have any proofreading capabilities to detect wrong unnatural amino acids? For sure. Particularly, yes, there are a lot of them. So the ribosome has size, recognizes the size. So there are certain amino acids whose side chains are just too large to fit in the A site, and they're rejected by the ribosome, even if they're charged onto tRNA. Backbone amino acids that have modifications like N-methyl groups, D amino acids, beta amino acids, the ribosome has trouble with those. Now, there are ways around it, around some of those, but the ribosome definitely has trouble with non-canonicals. A standard side chain modified amino acid, the ribosome has less trouble with, but certainly you have to think about EFTU affinity as you work through that as well. So, all right. I think I'm down to my chat list. Anybody else with questions?
I think that, yeah, that's all the chat questions. I just want to say someone already asked the question that I wanted to ask, but it's really cool work. Do you think we'll ever be able to, that's completely guessing question, but will we ever be able to make a completely non-degenerated code, like translate 60 amino acids?
Yeah.
Or there's something inherently.
Yeah. I think it could be done with the right ribosome mutations. I think it could be done. I'm not sure how to select for a ribosome that can do that. That would be a cool project, but I mean, the GC-GU issue is definitely going to be the hardest to get around. And you know, these hyperaccurate ribosomes that we pick were just something out in the literature. There could definitely be better ribosomes. There may be better ribosome mutants out there that people haven't looked at before. That could be pretty well screened with our assay. So if anybody has mutant ribosomes out there that they want to send to me, I'm happy to throw them into my assay and we can see if they work any better than those.
Yeah, most of the work that I've seen in using platforms like RISE are for more promiscuous ribosomes to be able to translate more weird stuff.
Yeah, that's right.
But I think we need both. We need promiscuity for amino acids, but really high fidelity for anti-codon.
Yes. Yes. Yeah, exactly. That's right.
You have another question in chat, I see.
Oh, yeah. Do you have any thoughts on the use of a tethered version of mutant S12 to further increase fidelity, or is that irrelevant? So I think you're thinking of Jewett's tethered ribosomes. I think there's some merit in that. I mean, this is a ribosomal protein, so the mutation is not in the ribosome itself, it's in the ribosomal protein. So I think it would be easy to add on to any common ribosome mutants. And those tethered ribosomes are nice for evolution, for sure, because you get both subunits together. So yeah, there's some merit in that.
All right. Any other questions? If there are no more questions, I want to say thank you, Matt, again, and thanks everyone else for a great discussion.
Yep.
And
thanks.
Have a great week.
Yep. Thanks for the invitation. Thanks for listening, everyone.
Bye.
Bye.